In this lesson, we'll cover the concept of pharmacodynamics. If pharmacokinetics describes what the body does to a drug, pharmacodynamics describes the opposite. In other words, in the study of pharmacodynamics, we look at what a drug does to the body. When a drug enters your body, it starts interacting with receptors, and this interaction produces a signal. That signal then travels through a series of biochemical steps until it ultimately results in some biological effect. So, for example, the signal can tell a gland to release a hormone or tell a blood vessel to relax. Our cells contain many different types of receptors, and each one, when stimulated, can produce a very unique response. The receptor types that matter most for pharmacology can be grouped into four categories: ligand-gated ion channels, G-protein-coupled receptors, enzyme-linked receptors, and intracellular receptors. Let's go through each one. We'll start with ligand-gated ion channels, since this is probably the simplest of the four to understand. A quick reminder, a ligand is just a molecule or ion that binds to a receptor. This type of channel has a binding site for its ligand, and when that ligand attaches, the channel briefly opens, allowing ions like sodium, potassium, calcium, or chloride to flow across the membrane. That flow of ions is what changes the electrical state of the cell. A drug like nicotine works exactly this way. It binds to a ligand-gated channel and causes ions to rush in, which is part of why it has such a fast, noticeable effect. Next, we have G-protein-coupled receptors, sometimes called 7-transmembrane receptors, because the protein winds back and forth across the cell membrane 7 times. These receptors are attached to a complex made of 3 parts: an alpha, a beta, and a gamma subunit, together known as the G protein. In its resting state, the alpha subunit holds onto a molecule called GDP. But when a ligand binds to the receptor, the alpha subunit trades that GDP for GTP, and this swap causes the alpha subunit to break away from the beta-gamma pair. Both pieces can now go on to activate other proteins inside the cell, setting off a chain reaction. There are 3 G proteins worth remembering. The first, GS, stimulates an enzyme called adenylyl cyclase, which produces a molecule called cyclic AMP, one of the cell's most important messengers. The second, GI, does the opposite. It blocks adenylyl cyclase and lowers cyclic AMP levels. The third, GQ, activates a different enzyme, phospholipase C, which produces 2 messengers of its own, DAG and IP3. DAG works similarly to cyclic AMP, switching on enzymes called protein kinases, while IP3 triggers the release of calcium stored inside the cell. One more thing worth knowing about G-protein-coupled receptors, and enzyme-linked receptors too, is that they can amplify a signal. A single activated receptor can switch on many enzyme molecules, which each produce many messenger molecules, so one small binding event at the surface of the cell can turn into a large response inside it. Moving on to enzyme-linked receptors. Like G-protein-coupled receptors, these have a binding site on the outside of the cell where a ligand, often a hormone or growth factor, attaches. Most of these receptors work through an enzyme called tyrosine kinase. When the ligand binds, it causes 2 receptor molecules to pair up, and that pairing switches on their kinase activity, which uses energy from ATP to attach phosphate groups onto each other. Once phosphorylated, the receptor becomes a docking site for other proteins inside the cell, and their attachment sets off the next stage of the cascade, ultimately producing the cell's response. Insulin works through a receptor exactly like this. The last type is the intracellular receptor. Unlike the other 3, this receptor sits entirely inside the cell rather than on its surface, which means the ligand first has to cross the cell membrane before it can bind. This is why intracellular receptors typically respond to small, fat-soluble molecules like steroid hormones. They're able to slip through the membrane on their own. Once the ligand-receptor complex forms, it can travel into the nucleus, attach to DNA, and switch specific genes on or off. Because this process depends on making new proteins, it's also why steroid-based drugs tend to take longer to kick in compared to something acting through a surface receptor. Cells don't just leave their receptor numbers fixed. If a receptor gets overstimulated for too long, the cell can pull receptors out of the membrane and recycle them, lowering the total number available. This is called downregulation, and it makes the cell less sensitive over time. This is actually why abruptly stopping certain long-term medications can be risky. The body has adjusted its receptor numbers to the drug being there, and removing the drug suddenly leaves it under or over responsive until things rebalance. On the flip side, if receptors are chronically blocked or signals are unusually weak, the cell can respond by inserting more receptors into the membrane, upregulation, which increases its sensitivity again. Now let's look at what happens as we increase the dose of a drug. As concentration rises, the pharmacologic effect increases too, until eventually every receptor is occupied and more drug doesn't add anything further. If we plot that relationship, we get what's called a dose-response curve, and from it, we can read off a value called EC50, the concentration needed to produce half of the maximum possible effect. EC50 tells us how potent a drug is. Let's say we're comparing two drugs, drug A and drug B. If drug A reaches its EC50 at a lower concentration, that means less of it is needed to get the same effect.
So drug A is more potent. But potency isn't the whole story. Look at the top of each curve. That's the Emax, the maximum effect a drug can produce no matter how much you give.
This brings us to the idea of intrinsic activity, a drug's ability to actually produce an effect once it's bound. If a drug binds a receptor and produces the maximum possible response, comparable to what the body's own natural signaling molecule would produce, we call it a full agonist. Interestingly, even with no agonist present at all, a small fraction of receptors, often cited around 15%, still show some baseline activity on their own, known as basal activity. A partial agonist, by contrast, binds the receptor but can never reach that maximum effect, even if every receptor is occupied. And then there's the inverse agonist, which binds the receptor and pushes it toward its inactive shape, actually reducing activity below the baseline level. On the other end of the spectrum are antagonists, ligands that bind a receptor and block it, reducing whatever an agonist would otherwise do. When an agonist and an antagonist compete for the exact same binding site, we call that antagonist a competitive antagonist. Its signature effect is shifting the agonist's dose-response curve to the right, meaning you now need a higher concentration of the agonist to reach the same EC50, so its apparent potency drops, even though nothing about the agonist itself has changed. Some antagonists don't let go once they've bound. They form a permanent, irreversible bond with the receptor. Because the agonist can never displace them, no matter how much you add, this lowers the maximum effect, or Emax, that the agonist can achieve, a true drop in efficacy. A separate category, the allosteric antagonist, doesn't even compete for the same site. It binds somewhere else on the receptor entirely and changes its shape so the agonist can't activate it properly. Like irreversible antagonists, these also reduce Emax while leaving EC50 unchanged. The last concept worth covering is the therapeutic index. No two people respond to the exact same dose of a drug in exactly the same way, so we need some way to measure how safe a drug actually is across a population. That's what the therapeutic index gives us. It's the ratio between the dose that causes toxicity in half the population and the dose that produces the desired effect in half the population. If you plot both curves, one for effectiveness, one for toxicity, you can find two key values, ED50, the dose that works for half the population, and TD50, the dose that's toxic for half the population. The wider the gap between those two curves, the safer the drug is considered to be, since there's a larger window where you get the benefit without the risk. And that's pharmacodynamics, how a drug, once it reaches its target, actually produces an effect in the body. Thanks for watching, and I hope this helped make the concept click.